Hello everyone, so we're back. It is day 2. Our blank cells have been growing for about 24 hours now at 37 degrees in our little DNA playground here. I've grabbed some of the items from the fridge. So these are the 3 plates. I'm just going to set these out. I usually bring them out at the very beginning of the experiment so they warm up a bit. Because as we go through the exercise today and get our engineered cells, when we pour them on the plates, there's quite a bit of liquid, and this will make the plates dry a lot quicker, as you'll see. Okay, so today we're going to be engineering our blank cells, and so we don't need the plate stencil, but we do need the loops, and we do need the bag of transformation buffer, recovery media, and we're doing raspberry red today. Okay. All right, let's have a look at our cells. All right, there we go. So you can see we got individual colonies. I must have touched just barely within the tube. You can see lots of cells on the first line, less on the second zigzag, and then the third zigzag must have been pretty much this single bacteria here. And this is more than enough cells. We're only going to use maybe 2 of these colonies. All right, so this is great. Let's move all this. We won't need this incubator for a little while now, so I'm just going to close it up, hang this up, turn it off here. And what I am going to turn on, though, is the cold station. Okay, so the first step in this process is getting the transformation buffer from the baggie here. That's going to be the red one. And transformation buffer is basically salt water. It has lots of calcium chloride in there, and the calcium really helps the DNA to interact with cells and helps the DNA to get into the cells. And it's really important that we keep this cold for most of the transformation process because it's quite harmful to the cells, and if you just put the cells in there at room temperature, a lot of the cells will die and your experiment probably won't work. So I'm going to put this into the cold station, which is going to keep it down at around 4 degrees Celsius. Okay, so I'm going to put that in there, and it'll cool over the next few minutes here. And then let's quickly go through these other 2 tubes. We've got the recovery media, which is going to be used right after we get the DNA programs into the cell, and it's sort of like the growth media for the petri dishes that we used, except there's no agar in it, and so really this will help the cells start growing and dividing again. And then we've got our DNA, so we're going to be doing DNA raspberry DNA program, and you can see it just looks clear. DNA is very small. It's a molecule, so you can't see with your eyes, especially when it's in water. You can check out a DNA extraction kit on the Amino Labs website, which will actually help you to visualize the DNA from fruit, for example. But in this case, you can't see that DNA because it's so small and because it's in water. Okay, and to help you try and visualize this in your mind, like what do these DNA programs look like? So they're actually really tiny loops of DNA, and so you probably have seen the double helix, the DNA double helix, right? Now just imagine that as a loop, okay? And they're quite small loops of DNA that are only large enough to contain the program or the gene for creating this red pigment, for example. And then there's also another gene in there which gives the cells antibiotic resistance. You'll probably remember that yesterday when we made the plates, we had some selective plates and we had a non-selective plate. The non-selective plates had no antibiotics in them so that we could grow our blank cells. And remember, blank cells are not engineered, so they're just normal bacteria. And then we added antibiotics to the 3 other plates. And the reason is because this plasmid has the antibiotic resistance gene. Once that plasmid goes into the cells with that plasmid then become resistant to this antibiotic, and only those cells will grow on our plates, on our selective plates. Okay? And so that's why you call it selective plate versus a non-selective plate, because you're able to select for your engineered bacteria using antibiotics and an antibiotic resistance gene. Okay? Okay, and yeah, that's good. So while we were talking about that, the cold station is now down to 4 degrees, so the tube is cooling, and so we can actually get on with the experiment here. So the first step is to collect some of our blank cells so that we can program them and put them into the transformation buffer. Okay, so this is pretty straightforward. We're going to just pop open this, get it ready. I'm going to open the tube here. Set the, whoops. And always remember to try and have the liquid in the bottom, and you might need to do the whippet to get it down. Alright, so now I've got my little blue loop, the small loop, and I'm going to collect, all I need is 2 of these colonies here, so I'm just going to drag, and actually this is probably more than enough. And you can see there's just a small white residue on there, and that is plenty of cells. Now we're going to go ahead and put that in, and you try not to touch the side of the tube, just go right to the bottom, kind of like you're playing the little game Operation, and then we're going to just spin this for about 20 to 30 seconds. Okay, and while we're doing this, the cells are being dislodged from the loop, and we're also mixing them and breaking any clumps apart. And the goal here is to have individual bacteria floating around so that they can interact with the DNA plasmids, and this increases your chance of getting plasmids into the cells. If you have lots of clumps of cells in here, so if we just stop for a second and have a look, we'll see that it is cloudy compared to just the regular liquid, so you could definitely have cells in there, and actually there's not too many clumps at all. I'm going to give maybe 4 or 5 more seconds of spinning here. And you kind of have to do it a little vigorously, as you can see. If you go really slow, you're not gonna mix the cells in at all. All right, let's see here. All right, that looks great. So now I've got some blank cells inside of the transformation buffer. And then the next step is to get another blue loop, and we're going to use this to get 1 microliter of DNA from the tube. And this is really easy as well. You're just going to put this in, and you're going to spin it for a second just to make sure that the liquid gets into the loop, and you can inspect it, and if you look closely you'll see the center of the loop has liquid in it. And then you're going to same thing right down the center, give it a quick spin here, and after about 5 seconds of spinning it should be all mixed in. Okay, that's all done. I'm going to put this in my discard container. I'm going to put the lid back on this DNA, and I'm going to put the lid back on my transformation buffer, cells, and DNA. And now we're going to wait 5 to 10 minutes, probably closer to 10 minutes. And during this period, while this is waiting on cold, the DNA is going to start interacting with the cells. It's going to start loosely binding to the surface with the help of calcium. And then after this 10 minutes, we're going to do a heat shock. And that's when we move the cells from cold to warm back to cold. And during that warm period, a lot of DNA can enter the cells. And then we put it back on the cold to trap the DNA inside. Okay, so what I'm going to do is I'm going to put the hot station to the shock, and it's going to maintain at about 42. It just happens to be there right now because the incubator was cooling. But yeah, we're going to need the shock for this next step. And of course, make sure the cold station is on because we don't want this to warm up. If it warms up, we're going to dramatically decrease our chances of success. Okay, so I'll be back in about 9 minutes. Okay, we're back, and it's been 10 minutes. We've had our cells, DNA, and transformation buffer chilling on ice, and now it's time to do the heat shock. So I'm just going to transfer it over and reset the timer. You can see the hot station is at 42. It's a good heat shock temperature. Restarted the timer. And now we wait 90 seconds. And during this 90 seconds, the cells are warming up, and the membranes of cells are kind of fatty in a way, if you've heard of lipids. And so when they're cold, they actually are quite rigid. They really kind of bind together really well. And when they're warm, they kind of vibrate a lot more. They're more fluid. And so as we're heating the cells up, their membranes become more permeable. And this helps to allow DNA to enter the cells. Okay. So we can't really see much right now, but there is a lot of activity down there. And we're at 1 minute and 5 seconds, so we got about 20 more seconds till we move it back. Alright, 1 minute and 30, and back to cold. So as we move it back to the cold, the cell membranes are going to cool down, become more rigid, trap some DNA inside, and on top of that, it's going to help stabilize the cells and keep them alive. You don't want to heat shock it too long. If you heat shock it too long, you're just going to kill all the cells. So we're going to just reset this, and I'm going to let it sit for 2 more minutes. And then after those 2 minutes, we're going to get our recovery media, and we're simply going to pour the recovery media into the little tube of cells and DNA, and that's going to dilute the salts, so that's not so harmful. It's going to give the bacteria food and nutrients to start growing again. And we're going to put once we do that, we're going to put the cells onto the 37 degrees hot station. So I just changed the temperature to 37, so it's going to cool down a bit. I'm also just going to put this tube of recovery media into the hot station, and that'll pre-warm it. That way when we pour it into the cells, then it will warm it up a little bit faster, give our cells a little bit of a edge, a little bit of a extra advantage. It's been about 1 minute and 10. Typically we suggest 3 to 5 minutes. So it was about 30 seconds when I restarted the timer, so I'll just wait another minute and a bit. And after, we're only going to need 3 more yellow loops, and we can call it a day. I'm going to put the DNA back in here, just in case. If you if you want, you can collect these, so the DNA lasts a reasonable amount of time, probably a few months to a year, depending on your fridge temperature. And so if you go through this experience and it worked, or even if it didn't work and you got to try it again, you can order different colors, and you can collect a whole bunch of different colors, and have a look on our YouTube channel. You'll see some other videos with some experiments where you mix plasmids, for example, and you can do that experiment. Okay, so it's been 2:40 on here, and I just reset the timer a little bit behind, so we're at about 3 minutes. So now we can go ahead with adding the recovery media. And so we can open up both of them. Come on, this one's tight. There we go. And what I'm going to do is I'm just going to pour it in. Okay, I'm going to give it a tap. And now that that's in there, I can go ahead and give it a shake. Okay, and when I shake it, it's just going to mix all the cells in and the transformation buffer in with the growth media. It's all going to mix together and become a much better environment for the cells to grow. Okay, so, and then after you're done, just give it a whippet and then put it into the hot station. And now we're going to let it sit for about 45 minutes. And during this 45-minute period, the cells are going to start growing again. They're going to also start creating their antibiotic resistance. Okay, because the next step after this, we put it onto the selective plates, which have antibiotics. So we definitely have to give them a little bit of time to start generating their antibiotic resistance properties. Okay, so I'm going to reset this, and I'll see you in about, actually one more thing. So it's always good every 15 minutes or so to come back, grab the tube, just give it a shake and a whip it, and put it back on. And this will just make sure it's fully mixed. There's lots of air inside of the tube so that your cells can grow well. Okay, so I'll be back in about 45 minutes. Oh yeah, and you're not going to need the cold station anymore, so I just turned it off. Alright, so you can go ahead, turn off the cold station. All you need is the 37-degree hot tube station for now. Okay, we're back. So it's been almost 45 minutes, and we're nearing the end of the recovery step, and we're on to the plating step, which basically means we're going to put stuff onto plates, namely our selective plates. So I'm going to get those out here, and we'll talk a little bit more about them, and then we'll get on with it. Okay, so one of the plates is a selective plate, so it's LB agar, and it has a negative sign on it, which stands for negative control. Okay, and a negative control is a control, an experimental control, where you're basically trying to remove something from the system to help you understand in the future, if something doesn't work, what might have happened. And so in this case, our negative control, one of the things we're trying to understand is, were these plates made properly? Okay, were there enough antibiotics? Did I mix them properly? Did I heat them enough? There's a lot that actually goes into making the LB agar plates. And let's say tomorrow our experiment doesn't work or something weird happens, a negative control will help us to troubleshoot. Okay, and so what the negative control is going to be for this experiment is, we're going to take some of our blank cells and we're going to plate them onto this selective plate. Okay, and the reason why it's a negative control with our blank cells is because they're lacking the DNA plasmid with the antibiotic resistance. Okay, and so what we would expect to happen if these plates were made properly is nothing will actually grow on this plate, because the cells will all be killed, or they won't grow. Their growth will be prevented by the antibiotics. Okay, and that could tell us a lot. Like, let's say tomorrow we got a lot of growth on our plates, like a crazy amount of growth. It was just kind of a blur, a film, and we're like, what the heck happened? They're supposed to just be raspberry red colonies. We could look back at the negative control, and if there was a lot of growth of this bacteria, of the blank cells, that would tell us that there perhaps wasn't enough antibiotics in there, and that's why we had all this weird growth. Okay, so that's our negative control. It's going to be blank cells. Now we have a positive control. A positive control is sort of the opposite. You're sort of you're doing something that you're pretty sure should work, right? If everything is right in the plates, it should work. And in this case, I've got a plate of purple cells. If you look into your kit, you should probably see a small tube that says positive cells on it. These are cells that already have a very similar plasmid to any of the colors that we offer. It's a chloramphenicol resistance plasmid. So we're going to use this plate, and in that case, if you have the tube, you're just going to dip the loop into the tube just like you did with the blank cells and smear it over the plate. And so the idea here is that if there is the proper amount of antibiotics, if there's nothing else weird happening, these cells should without a doubt grow, because we already know they grow on chloramphenicol. Okay? And then we get to our experimental, which is our sample from today. So we're going to pour some of our sample onto the plate, and we're just going to spread it all around, and hopefully tomorrow we get some, at least one red colony. Okay? So that's what we're going to do. It's been 47 minutes, and that's ample time for recovery. You can let it recover longer if you like. You can let it recover for a few hours or even overnight. That's up to you, but 45 minutes is plenty. You could even do some experiments of your own, see if 30 minutes works. Okay, so let's go through and just do this in order presented here. So we're just going to touch a colony. We're going to get our negative control, and we don't need to worry about streaking. We're just going to spread them out. So you can see there's lots of cells up here that I just put on. We're just going to rub that all the way down. And I'm going to leave the lid off just to make sure these dry well. And I'm going to put this in my my little discard bin for now, but then we'll put it all in the inactivation bag later. I'm going to do the same thing with the purple bacteria here. We're just going to touch a colony. Isn't that a nice purple? That's our purple color. See, it's quite beautiful. It's one of my favorites. Okay, so we're going to spread that around, same. And I can see a little bit of liquid on there because we put the plates in the bags right after, so there's going to be a little bit on the surface, so you can leave the lids off, let them evaporate a bit. Alright, and now we're on to the last one here. We're going to try and pour about half of the liquid into this plate. If you pour all of it, it's just going to pool like crazy, so just try and do a couple of drops. That should be plenty. And same thing here. Spread it out. Okay, so you want to go at a very sharp angle. Okay, like you want to be almost horizontal over the plate. And I'm just going to smear it around gently, not to puncture the agar, while I spin it. And this takes a bit of practice. But sort of like, can you rub your head and your belly at the same time? Okay, so that's well spread out all over the plate. And all of these are a little wet, especially this last one. As I mentioned earlier, this is the main plate you want to consider drying before you put into the incubator, because again, we're going to put the lids on, then we're going to flip them when we put them in the incubator, so it's always really important to make sure there's no pooling liquid. So just like the blank cells, I'm going to let these sit for a few minutes, probably 5 minutes we'll try, and reassess at that point. In the meantime, I'm going to turn off the hot station, and I'm going to turn the incubator on to 37, because that's what we're going to be incubating these plates at overnight. Okay, so I'll be back in about 5 minutes. Alright, so we're back after letting the plates dry a bit, and I had a look at the positive and negative, and they're decent. You don't see any water drooping as you spin it. I don't know if you'll be able to see this, but there's a little bit, so there's a bit of pooling at the bottom that you can see. If I were to just spin this, you might see liquid slowly falling still. It's much less than we had, but it's still something. So what we could do is we can actually, to accelerate this, we can open up our incubator here. I'm just gonna set the plate in with the lid off. I'm gonna move all this other stuff out of here. Oops. Remove all this. I'm just going to set this in the incubator, top up and open. Close this for only a couple minutes. You don't want to close it for too long. Wait 2 more minutes or so. Let's see what it looks like after that. Okay, be right back. Okay, so I'm back here right after a couple of minutes of letting it just our open plate stay in the incubator. Let's have a look. Definitely don't want to leave it in there. Wow, look at that. The tiniest little bit of liquid coming down. I don't even know if it's going to make it to the bottom. Oh, there's still a little bit around. Let's put that in for another couple minutes. But you get the principle. Really, it's just a matter of making sure there's no liquid that's going to go onto the lid once you flip it. So we'll be back in a few more minutes and we'll just finish this. Okay, we're back, and the plate is quite dry. There's no liquid pooling, which is great. And now it's just a matter of us incubating it and seeing how things go. So just like before, let's stack all of these up together. So again, we've got our experimental plate, our negative control plate, and a positive control plate. We're done with these. We're just going to move them over here. So you get your paddle. I'm going to just zip these on. I'll put this little puck on, and then we're good to go. Into the oven. Close the door. Great. Now I'm going to reset this timer and let's see how it grows over the next 24 to 48 hours. Okay, so I have the bag from yesterday. I'm going to put our tubes in here. We've got one. I've also got the engineered cells. I'm going to hang on to this, and you'll see why shortly. And then, yeah, our non-selective cells, our blank cells, can also go in there. We can hang on to our DNA, as I mentioned, and we'll inactivate all of these things in a couple of days once we see what our results look like. And you can put the DNA back into the fridge, and hopefully you can use it for future experiments. Alright, see you in 24 to 48 hours.